can do it really interactively. It's a lot more fun if the points and the exchange are coming also with the public. But I think we've thought about a number of topics that are covering some of the the topics that were brought in during the different presentations. And so, what shall we start with?
I thought that Nadia was going to animate us. What would you like to start with?
Well, I think one of the themes we really started by discussing was, that there are obviously a couple of themes that ran through this. One was taking, very rigorous and precise methodologies, but another was focusing on the size of data sets. And actually, our last two very rapid neighbor joining variants really illustrate that extremely well. And of course, one of the things we were discussing when we were thinking about this panel was a fundamental challenge now with data sets being so large is they are moving beyond human experience. We can't, the human eye is, even in the era of AI, the human eye is an amazing pattern finding machine, the human eye, the human brain. We can't do that anymore. And so that's, I think, maybe, do you think that would be a good place to start?
So maybe, one question to the audience. I mean, what one of the challenge with the large data sets, I mean, in principle, like in statistics, you would expect having more data leads to smaller confidence intervals, where like better, less uncertainty. But it feels like so far we haven't seen like lots of cases where really harnessing very large amounts of data, like enabling like biological insights that were not possible so far. And so that's a question maybe, to all of you, like, do you have a sense why that may be? I mean, when you're developing some tools that are able to cope with, like, here, I don't know, you had 100,000 sequences, do you see some applications that could really take advantage of this large amount of data? Or conversely, I mean, and that is like to Ed's point, the systematic error, the model error does not go away. We've seen sometimes, if the data could be highly correlated. That was the example of Yanis with fragments in many, many genomes because they were all annotated from the same flawed reference. Any thoughts maybe on these emerging difficulties and opportunities, but also challenge of very large data sets?
I guess what.
Yeah, but go ahead, Lars, and Fiona has some thoughts.
Yeah, so I immediately start thinking about visualization, and when Inan had his talk, one of his limitations, I believe, was being able to visualize things, right? And with the large trees, you basically have the same issue. There's no way I'm looking at my very large trees, and I know people who figures visualization is the next frontier to many things. And I am guessing that what you would want is visualization very strongly connected to things that you decided to compute, and what that would be would be something new, I guess.
This makes me think that we should have perhaps like a joint session with the visualization cozy, right? There's another session that is probably they're also really interested to know where there's some needs for more visualization.
Wow,
Fiona,
that's
go
a
ahead.
fantastic idea. I'm joining in. I been going between sessions, so I came into this one. I'm so pleased to see you talking about like the big picture messages from this, because I feel like I'm getting a lot from this. But I just want to say, somewhat naively though, is when I'm dealing with challenges like this, I tend to feel like I gotta focus on the use cases, and that I think the problem is we have so many different use cases for a tree, and so it would be good to think about certain particular use cases and analysis, visualizations, I think is what I was going to bring up too, for that use case, but also just simply that ability to be able to go through, a tree in different ways and look at it in different, flexibly and interactively in different ways, I think is really important. But I think it's really important not to lose sight of the basics, right? I'm going to be talking at the Quest for Ortholog meeting tomorrow about just the importance of remembering ultimately this is about characters and looking at a bunch of characters and just making sure that that robustness is there as we get to really big trees, which is a huge challenge. That's fine.
Well, thank you for the teaser.
Seth, do you have comments on big data?
No, I actually have a technical question. On the record, would you mind giving me permission to record and share this recording?
Yeah, of course.
We would be delighted.
That's all I needed. Thank you.
Excellent.
All right, the point about big data. So the running discussion in my group is if the student or staff members complain about how big the data is, I tell them you are in the wrong business. So that's on one hand. On the other, garbage in, garbage out, right? So the quality of sequencing instruments, the primary source of data that we're analyzing has been improving, which means if we look at the older data, it's garbage, right? But there's a lot of information built on that poorer quality data. I shouldn't call it garbage. I'm just poking the nest here, but you get what I mean. So the quality is improving, but we always build upon what came before us. So I think I'm taking two different positions here, two polar positions here, but we do have a good opportunity to bridge that gap using some of the emerging technologies like the ones we talked about today, or some machine learning methods to actually reconcile that. My 2 cents.
Yeah, thank you very much. Any other thoughts on the topic of large data sets?
Well, I have to be the curmudgeon. I think it's much nicer to get a paper accepted or to get funding if you say, I have this exciting new application and we're going to get brand new data, unlike data we've ever seen before, as opposed to I'm going to acquire new data carefully to check all the old data and do error correction and clean something up. And so, unfortunately, that bias towards the exciting new stuff doesn't help us manage the explosion of data, I think.
That's a good point, and it's true. It's surprising if you, for instance, look at the number of genomes that are, fully assembled. I mean, that number has been stagnating quite.
Like 3?
Well, I mean, okay, that's maybe an extreme position, but well, Danny's got very high standards. We knew that already.
If you're interested in studying repetitive regions, for example, you may, that's something where there may be real problems with assembly in most genomes, and for most applications it doesn't matter, but if you want to know what repeats are doing, it's a huge trap.
At the same time, there are some consortia like the Darwin Tree of Life that is now using routinely long read sequencing to provide really remarkably well assembled genomes. It's at the annotation level that there's still, I think, quite a lot of work to do there. Maybe also a point, I think, on this, maybe for the younger people in the audience, is also sometimes it may feel also a bit, if the data has been produced by large consortia, it feels like, okay, they must be really good, and because there's been so many people who are involved. but actually you'd be surprised sometimes how, the number of co-author does not necessarily correlate with the number of eyes that are looked at any particular subset of the data.
Yeah, I can probably add to that. Christoph has pointed out that the B10K data, and I've participated heavily in the Bird 10K consortium, that there are some issues with this, all of which I think we're aware of. These things, unfortunately, are always evolving, and if anything, I would say the annotation is probably one of the more ignored components. It's very easy once a sequence goes into, say, NCBI or ensemble or whatever, to not update it further. And these things should be getting new looks and new annotations.
So if you have any thoughts on this topic, feel free to come spontaneously. But I will jump on. Connect also with the talks we heard about transcripts evolution. Kind of also that of course is dependent on good annotation. But actually, I would argue in reverse. We've seen also as people have been trying to sequence deeper and deeper the genes and seeing all the isoforms. Actually, this is probably also an area where EvolCompGen can help, because the evolutionary selection of these transcripts is probably what is going to ultimately solve the issue of transcript annotation. At the moment, what we see is that anything that is kind of lab-based will, the deeper we sequence, the more isoforms we annotate, and that's even true for species like human and mouse. And so I think this is probably a topic that we will hear a lot more in the future in this community.
My advice to you is to only work on bacteria. Then you don't have to worry about alternate splice forms. Although, Fiona, you'll probably tell me there are all sorts of other pitfalls I'm unaware of. Retrats. Well, there is that. There is correction, though. There's also the experience of you write a paper about a gene in an organism, and by the time the paper comes out, the organism has been reclassified to a different clade. It has a new name, and the gene has been redefined, and 30 base pairs have suddenly disappeared. So that's, on the one hand, very frustrating for the person who's writing that paper. On the other hand, somebody is paying attention to the correctness of the data. And actually, gene sequencing has revolutionized our understanding of bacterial taxonomy. The extent to which things have been reclassified over the last 10 years is really, you have to publish fast before something changes its name.
So one other general trends we've noticed is that a few years back, we started having quite a lot of cancer phylogenies talk. And I'm not just saying this because Russell's in the room. Actually, we had quite a few, Katerina. Where's Katerina? Yeah.
She was in the room.
No, you're there. So we had, and so I wonder, this seems to have slowed down a little bit. It would be really interesting to have your insight. And it seems like what we've now started to have more of the lineage trees, fueled by the kind of single cell transcriptomics revolution. So I think it's quite interesting. We had a few talks on this, but maybe like for those of you interested in this, in the case of the lineage, is not really evolution, more like trying to recapitulate like the cell division process. In the case of cancer, it is an evolutionary process, but people often don't think of it really that way. These emerging or other applications of trees and phylogenetics. I don't know if there are any thoughts that you'd like to share on this. Any sense why we have maybe an apparent slowing down of cancer phylogenetics? Methods.
Well, I can comment. I don't think there actually is. I think what's changed is that it's become so accepted in the cancer community that people are presenting it there rather than in the phylogenetics community. But there is a need for a lot more feedback because there are hard problems. And you talk about what to do with bigger trees. I think, what do you do with somatic evolution on 1 million single cells repeated over 1 million people? That's an interesting problem that needs better algorithms that will come from the phylogenetics community, much more likely than from the cancer community. But I think there's also a lot of interesting feedback we had the other direction. And there's the old question about reproducibility of evolution. You only get to look at the species tree once for the planet, but with cancer, you could look at the same evolutionary landscape being navigated 1 million times in 1 million different tumors, and you start to see interesting things about reproducibility or non-reproducibility of the process. I think there are a lot of good questions still.
Alternate universes.
Well, thank you, Russell. That was really, really insightful. And I think we are quite happy this community to be really at the cutting edge. And then once things become mainstream, they go to other communities.
Yeah, although an unfortunate thing perhaps is the loss of the potential for cross communication, as you pointed out, among, for example, cancer phylogenetics and organismal phylogenetics and so forth, and potentially other areas like immune cell development and so forth. I think the potential for cross-fertilization is very real because they're very similar problems, but not identical. And I don't know what all of you out there think. I assume that high-level evaluation is hopefully agreed upon, but, I don't know what other thoughts there might be about specific cross-fertilization.
I mean, yeah, the concept, for instance, of a mutation tree, which we've seen also in this session, is something I think that was not. It is an innovation of the cancer phylogeny field, because that turned out to be really important. In the end, you don't care so much about the exact placement of like that particular cell, but you want to know that particular mutation happened maybe after another one. And then I seem to recall during COVID, where you had lots of identical sequences, all of a sudden that became also quite relevant. So I think indeed, like it's every new application, back to Fiona's point, is kind of driving probably a different lens on the problem, and then potentially new concepts.
Well, you have the related question of sequencing a population in planktonic culture or something like that, but you sequence day after day, and so then you're able to pick up mutations in that population and plot how they, do they spread, do they not, do you need to have this mutation before that one? So that's not trees, but it is also a very intensively data-driven approach, computational approach to looking at what the relationship is between the causal relationship between changes and other changes.
So to connect with another talk we heard from Lucy, and I think we have also, in terms of conceptual advances, the field of phylogenetic networks, is also one that has been quite vibrant. We've not had many talks this year, but it seems this was the genesis for Lucine's new compact representation of trees, right? Which is like, we've all been using the Newick format for forever, and so this is really interesting, kind of a new approach, which is then directly also applicable to machine learning. So it seems this community has been quite let's say resistant to the hype around artificial intelligence, perhaps more than some of the other sessions we've had. Some talks. Do you have any? In particular, the bootstrapping. Yeah.
No, I mean, how did we get from networks to,
Well, this is, I'm trying to cover like different topics. So please feel free to chime in to,
may I comment on networks
Yeah,
briefly?
sure.
So within the sort of evolutionary biology community's approach to computation and systematics, there's been a lot of attention to network bio, in terms of hybridization. And the evolution meeting is going to be here in Montreal as it happens in 2 weeks, and they're running a whole symposium on networks. So that's, maybe we want to find more ways to reach out to that community. And if you're interested in phylogenetic networks, you probably also want to look at those journals and see who's publishing what.
Yeah, that's a really good point. So those of you who are staying a bit longer in Montreal, there's more.
We're never going home. I have a pitch, which is increasingly we're having new experimental techniques that make it much easier to study non-mobile organisms. So with next generation sequencing, you're able to just sequence a cell without having to grow up lots and lots of cells in order to have enough DNA to do Sanger sequencing. And I think that our understanding of biology has really been kind of skewed by the fact that it's been easy to study a small number of species, and it's easy to think that, all of bacterial life behaves like E. coli, for example. And so I think there are exciting questions to look at in terms of how can we use those sorts of data to understand things outside of the standard model organisms, and people might look for opportunities to work on questions around that.
So
Paul.
So yeah, now for something completely different. No, I actually was intrigued. So sorry, so my comment was actually spurred by something Christoph said, right? Which is, if you go around to a lot of the different talks here, it's clear, right? AI ML approaches are, you see everywhere. Now we got to see, there's a very nice talk on, Bootstrap. Being something maybe amenable to ML, but I actually came here expecting to learn about how people were, as we all know, right? Evolutionary modeling is at the core of biology, right? Evolution is everywhere. And so I expected to see people really thinking about how to apply ML approaches or incorporate ML approaches into phylogenetic reconstruction, or figuring out how to sort of teach machines to understand evolutionary reasoning, right? There's some really simple concepts. I didn't see anything like that, but maybe others did. And I think this would be actually a really interesting thing if people have perspectives on, the potential or ideas that you've had, or maybe things that I didn't see in this session, that bears on ML in evolutionary reconstruction or evolutionary reasoning in general.
So thank you, Paul. I'm bouncing this question back to all of you. And if not, I'm sure, it may also be one of these topics where we need a little bit longer, and then next year we may have more.
Perhaps I could interject something, though, just to be a contrarian, which is in sort of evolutionary and comparative genomics, we're really in a state where the black boxy aspects of machine learning may actually not be advantageous. If you devise a maximum likelihood model, the other ML, you're actually talking about events that can be described. You're either making an explicit approximation about how evolution worked, or you're actually modeling events. In parsimony, you may be trying to minimize numbers of events that are discretely describable as true biological events. And maybe in this field, machine learning will have limited applicability because we have ways of directly incorporating the biology into the algorithms and the mathematics. And I'm not necessarily saying that's true. I happen to believe it, and I'm putting it out there as a contrarian. What do other people?
Well, I'm with you.
Okay.
So I think we should wrap up
Yes.
on this point, because the keynote is going to start very soon, just in a few minutes. You will still have time to grab a coffee. But in closing, I want to thank you all for sticking in this session until the end. And I do want to make also a call if you would like to get involved in the organization also of the next EvolCompGen, which will take place in the next year, also in Liverpool. You can also even contribute, even if you are unable to join the meeting. Though of course it's always more fun if you can join. Then please get in touch with any of us, and we'll be very happy to welcome
There's
you.
lots to do.
Yes. Okay. Well, so thank you all for joining, and then see you next year.